This is the second prep for lecture 29. We're going to have several of these shorter ones because I really think it's better to have this sort of more digestible topics. And we're just going to look at the patterning along the anterior-posterior axis, and this is how that axis goes. And it turns out the signaling center is in the posterior of the limb, but and there's a gradient of activity from the zone of polarizing activity that is required to make this gradient that then leads to the anterior versus posterior specification. And one of the reasons we know that the zone of polarizing activity is a source of these sort of morphogens, I guess you'd call them morphogens, the different kinds of digits are forming in response to them, is you can do transplantation experiments. So if you have a limb bud and you transplant a large portion of cells that are from the zone of polarizing activity from another one, you will get mirror image duplication. And so let's just look at the normal situation first. So here's our polarizing region. And the idea we'll talk about, it turns out that sonic hedgehog is the morphogen, but it acts through some other molecules that we'll be discussing later. And you have this decreasing morphogen concentration, and then digit 4 forms in response to the highest level of that. So it's above a particular threshold, and then between the first and the second, thresholds, then you get the third digit forming, and then below the next threshold is the second digit forming. So you can go back to an earlier lecture right towards the very beginning of the semester where we talked about these, different gradient levels, and then the different thresholds that led to different fates. And there was a worksheet as well that you worked on in class to help you, play a little bit with the gradients and see how changing the gradients and the thresholds and so on was made a difference to the outcomes and the general shape. So the cell fates were the same, but they would be distributed differently depending on the gradient. So if we put enough of the zone, the ZPA in the anterior region as well, then what we have are two gradients that have the highest level of the morphogen in the most anterior and the most posterior, and then they grade away towards sort of the more medial central region. And when you do that, you get this perfect mirror image duplication with the outside having digits 4, and then within that digits 3, and inside that digits 2, right? Multiples of those, and with a plane of symmetry sort of right in the center like that. And so you can correlate this idea of these two gradients now and producing this mirror image duplication. And then if you just transplant a very tiny amount, you still get a gradient, but the level of signal is less, of course, because you have fewer cells giving rise to that signal. And it grades away and faster than here, and so it only extends a short distance. And what that will do is, this particular level of signaling, which is about here, is equivalent to digit number 2. So you just get the most anterior digits duplicated because the level of the signal is lower. Okay. And so the color coding here is for the different digits, but that also corresponds to the amount of signal on this gradient diagram here that gives rise to each of the different digits. So sonic hedgehog is the morphogen for that. It's expressed in a posterior to anterior gradient. You can take fibroblast cells, those are just cells, skin cells, and you can genetically engineer them to express sonic hedgehog. And then if you transplant them into a wing bud in the anterior, you get the exact same kind of result here as you would have gotten if you'd transplanted a zone of polarizing activity, which tells us that the key feature of this is the sonic hedgehog, right? And you can see a connection between the actual type of digit and the amount of sonic hedgehog. And there are extra digits that form when there's signaling defects in sonic hedgehog. Now, some of these things are similar if you use retinoic acid rather than sonic hedgehog, but we know that the effect of retinoic acid is actually to induce sonic hedgehog formation. So it is sonic hedgehog that's actually affecting the change in fate of the cells. Retinoic acid is just basically turning on the tap for the signal that then produces the gradient. If you there's a number of different mutations, and they have different names, but the ones that I know about, that I have read enough to understand how they work, are mostly regulatory mutations for sonic hedgehog. So they don't change the gene coding part, but what they do is they alter the regulatory sequences upstream, so they cause the gene to be turned on in places where it's not supposed to. And one of the types of polydactyly that results, you can see in this cat. It has extra digits, and they are called preaxial. So if you look at sort of like through the center of the hand, on the anterior side is called preaxial. So it's before the midline. And then there's also kinds of polydactyly that have an extra finger here, and those are called post-axial. So preaxial, and this is preaxial too, because if that's the midpoint, that's sort of the anterior and posterior of the limb, or sorry, of the hand, I guess. And if you look at the development of this, which they do in animal embryos, of course, not human, they find that the sonic hedgehog is expressed in more anterior regions than normal, and that creates this kind of situation. This is what a preaxial polydactyly would look like, with two extra just anterior digits, and then the more posterior ones are normal. And there are a number of types of cat that do carry this dominant mutation. It's dominant because if you have the normal copy of sonic hedgehog and then you have the copy that's expressed in the wrong place, you only need one copy of the mutant one to have the expression in the wrong place, right? And so, yeah, there are breeds of not breeds of cats, but polydactyl, certain populations of cats that carry this dominant mutation that leads to the polydactyl phenotype. So what that means is you have a series of cells, and they're receiving different levels of sonic hedgehog signaling. And you may have wondered about this kind of question before when we were talking about morphogens, because we didn't go through it in a huge amount of detail. But how do cells actually measure the amount of a signal? And it's because of the downstream proteins, the transcription factors, most of the time, that are activated. So if you have a small amount of binding of the signal to the ligand, then you will get a little bit of the transcriptional transcription factor going into the nucleus and turning on, or in some cases turning off, particular target genes. And if you have more signal, then you will have more of these target transcription factors being activated and turning on sets of genes. So that's kind of the quick answer. But there are 3 transcription factors, GLE1, GLE2, and GLE3. We're only going to look at GLE3. And so in response to sonic hedgehog signal, GLE3 in the cell is bound to a protein that's called suppressor effused, which we abbreviate this way. And so when it's bound that way, it can't be activated. But in response to sonic hedgehog signaling, the connection between the two is severed so that GLE3 can go in and act as a transcriptional activator. When GLE3 is bound to suppressor effused, it acts as a transcriptional repressor. And so if you go back and look at hedgehog signaling, you will see cubitus interruptus is similar to this. It's the same kind of signaling pathway. And this is before midterm 1. And so the level of sonic hedgehog signal is interpreted by the cell as the GLE3 repressor to activator ratio. So if you have no or low sonic hedgehog, this ratio is really high because it's almost all repressor and very little or no of the activator form. And then the more sonic hedgehog signaling you have, then the lower this number goes, because most of your repressor was converted. So this is high when you don't have much sonic hedgehog signaling, but this number drops with strong sonic hedgehog signaling. I have seen it mostly expressed this way, but some sources also express it this way. So, and that's going to be the opposite, right? So you have to remember what happens. This is high when you don't have sonic hedgehog, and this is really low when you don't have sonic hedgehog. And when you have lots of signaling, then this is high and this is low, right? So, and if it's expressed this way, then it's just, when you have lots of sonic hedgehog signaling, you have lots of activators, so this ratio is going to be high. And when you don't have much signaling, then the repressor is going to be more, so this ratio is going to be low. So just give yourself, learn it one way, but then just remind yourself that if the ratio is expressed the other way around, the numbers are opposite. So in the absence of sonic hedgehog, the receptor patched inhibits the receptor smoothened, and we have this is interesting. I was distracted for a second because I was watching. These are probably actin filaments and motor proteins. So your GLE, in this case GLE3, but there's also GLE2. We're not going to talk about GLE2. They are transported along these either microtubules or actin filaments to the surface, the tip of the cilium, and they walk back down and they get sort of circulated. When they are phosphorylated, they are targeted for cleavage in the proteasome, and the repressor forms of these proteins are produced. Okay, so things to pay attention to is the phosphorylation status, the fact that they're bound to suppressor effused, and that there is this kind of cycling of them kind of, around the cells. And then different genes, for example, Hox genes, which we aren't going to talk about in specific detail, but the different fates of the fingers anterior to posterior is going to be based on HOX genes. Okay, so these are our target repressors. When sonic hedgehog binds to patched, the receptor gets internalized, and then smoothened is free. It's not being repressed anymore by patched, and so it's able to inhibit or inactivate or cleave suppressor of fused from the GLI proteins. And they're not being phosphorylated anymore and targeted for destruction. So now they're intact, uncleaved, and they don't have suppressor of fused on them anymore. And so they are able to enter the nucleus and activate target genes, one of which is GLI1. Another of which is patched, which I think we mentioned briefly when we were talking about fly development way back when. So the key points here is that when sonic hedgehog is not present, these proteins are cleaved into forms that bind and inactivate, turn off the target genes, whereas when when sonic hedgehog is present, the smoothened receptor is able to remove suppressor of fused, and these proteins are no longer targeted for destruction. They go into the nucleus and activate their target genes. And the more of this you have, the more the posterior specific proteins are being expressed. Okay, so some of the evidence for the fact that sonic hedgehog, the main role it has in the specification of posterior versus anterior digits is the activation of GLI3, the formation of the active part. If you knock out GLI3 in mice, there are extra digits that are formed, and they all have the same appearance, so they don't look any different from anterior to posterior. So what that means is that the formation of digits themselves is not dependent on GLI3, but telling them what fate they're supposed to have, which digit they're supposed to form, is based on GLI3, because we lose that specification when GLI3 is knocked out. If you knock out sonic hedgehog in mice, you don't get any digits at all. And basically, this is because the GLI3 repressor protein is the only one, excuse me, the only one it has in the whole limb bud. So all the target genes are repressed. And then sonic hedgehog knockout and GLI3 knockout together, you get the same as the GLI3 knockout by itself, right? And that's because when you knock out GLI3, there's no GLI3 protein at all. Versus the difference between this one and this one is that the sonic hedgehog knockout means that there's no sonic hedgehog protein, so therefore the GLI3 protein, which is still there, is all repressor. So this must be the explanation for this part. But when we knock out both sonic hedgehog and GLI3, we get the same as the GLI3 knockout by itself, which means that the main role of sonic hedgehog is the activation of GLI3, right? Is its effect on GLI3. Because when you knock out both of them, they both knockouts have the same phenotype as just knocking out this, which is the target. Hopefully that makes sense. There's a logic to it. Okay, so sonic hedgehog is absent from the anterior limb bud, and that the GLI3 is all repressor there, and that should give us our more anterior fates. This is kind of a summary of how the whole thing works. The sonic gradient starts from posterior to anterior. You're going to have different ratios of activator to repressor all along, with the highest level of activator in the posterior region, and then in the anterior region, the very lowest, down to like 0 of the activator. Okay. The higher levels of sonic hedgehog and so on are needed for those posterior fates. And the knockout results that we just talked about suggest that the role of sonic hedgehog is really suppressing the repressor function in the posterior part and allowing for the activator function to be formed. Okay, and we know that there is some kind of self-organizing aspect to limbud, and the digits can form without either the activator protein or sonic hedgehog, but these guys are the main role that they have is to confer anterior posterior identity. There is some evidence coming up that I don't think I'm going to talk about in this prep about just the fact that cartilage can sort of coalesce on its own in response to some other signals, but I will probably not get a chance to talk about that quite yet. Okay, and so in discussing this data, I've mentioned this before, but one of the things I like to do is make some kind of little diagram. And so I've got the sort of the normal position, like this is how it works. Sonic hedgehog mediates the conversion of repressor GLE3 to activator, and then we get a normal digits with the proper anterior and posterior specification. And then I've just repeated the whole picture, but crossed out the parts where mutations have happened. So here's a knockout of sonic hedgehog. That means there's no protein being made. And what's the result of that? The result of that is you're not getting any conversion at all of GLE3 repressor to the activator. And what is the result? That leads to no digits, right? So this is just summarizing the results, and then you can kind of see the patterns, right? So if we knock out GLE3, the gene, it means there's no GLE3 protein at all, either activator or repressor. And you get too many digits and they're all unpatterned. And then for this knockout, sonic hedgehog, the double one, and this protein, too many digits all unpatterned. So the key thing that I used this figure for was to see what the difference was between these two. How come if you knock out sonic hedgehog and GLE3, you get one phenotype, and if you just knock out the sonic hedgehog, you get a different phenotype? The difference is this part. The fact that if you just get rid of, delete the gene or inactivate the protein in some way, you're going to have lots of GLE3 protein, and that's the difference between having no digits and then having too many digits all in pattern. This is because neither you're not repressing target genes, you're not activating target genes at all, because you don't have any protein, so that whole process is not being regulated. I hope that makes sense. Now, this diagram may not work for you. You may need to actually go back to that slide and go, okay, let me draw, and you can draw something yourself that makes some sense. I do think that working on these figures and making them, I know it will take you some time, but I think it really helps you understand. And the process of making this puts it in your brain so that you're not going to have to cram at the last minute so much, because there's already a place in your brain that's gone through and processed this in detail, right? This kind of thing. Okay, and so yeah, this is the spontaneous self-organization of digits. So I'm gonna I'll probably do this tomorrow. It's getting kind of late, but we'll start here in the next prep.